You know, one thing that I wanted to show you that I think is important, because remember I was saying that all science is a social activity, you know, it's human beings, and we exist in social world, and understanding new knowledge is not just the hard science, it's also like sociology, you know, and politics and all that other stuff mixed in, people's personalities. You know, I mentioned Rosalind Franklin, who's one of my heroes. You know, she was a great experimentalist, as we say. She was great in the lab. And she wasn't just bright, she was great. She had a great pair of hands, as they say, great in the lab. And she did work, here we go, extraneous stuff. She did work, she did X-ray, she was like the world's best X-ray crystallographer. And that's why her technical skills were one of the reasons why she could see the structure of DNA. And you know, so she didn't get the Nobel Prize because she had passed away, tragically, of cancer. You know, she did have challenges in her career. So she was a woman, and I did mention that, in King's College, London, which was a very male environment, but she was also Jewish, and in a very, well, I don't know, Episcopalian, whatever, Church of England environment. But, you know, it can be a challenge for a lot of people, but I don't know, we have no choice but to keep on trying. But there was another thing that I passed over, and I wanted to show you about understanding about how hard it is to see the real meaning of things, to see the meaning of things, to see into the future. So Linnaeus, you know, he was the first and foremost the botanist. He classified plants according to their floral structures, which we still do today, largely, and numbers of stamens and pistils. His work represented a great breakthrough. It is the foundation. It provided the foundation for evolutionary theory, but he himself could not see the implications of his own work. So I had prepared a slide to illustrate this, which I passed over in a rush on Monday. So one can only see so far through the fog. That is the sum of all current paradigms. This is really reality. I mean, it depends on what you're working on. If you're working on stuff that's fundamental and basic, this is what you're doing. You're trying to see through the fog that is the current view of things. And it's hard to do that. And so we are all ultimately limited by the vantage point of our own time. Now, why is this point? So this is Linnaeus, and he can only see so far through the fog. And then time goes by, and there's, you know, in the future, this person, whoever that happens to be. I don't remember who I put there. And that person can only see so far, much further through the fog, etc. We're all prisoners. And this is a relevant point to the subject of epigenetics and inheritance, because that's what we're faced with. We're faced with a model, a paradigm, genes, the genome, which is very powerful, the genomics. And you know, I've had people totally dismiss epigenetic inheritance as being unimportant. Prominent people, I mean, big shots dismiss it. And when a person who's maybe the leading plant geneticist says, this stuff is all nonsense, well, that does present obstacles, you know, to making progress. And none of us, no matter how brilliant we are, really see far. You know, the characteristic, I think, of people that I think of as great geniuses is like, there was a person, Barbara McClintock, I don't know if anybody of you have heard of her, corn geneticist, a great scientist. You know, there's a unique characteristic to really great scientists, and that is that they could see further. They really do see further somehow than others do. You know, it's amazing. So I just wanted to make that point. You know, and that's just a quick review. On Monday, turned out that there was a gene in the species that if it was methylated, DNA methylation, it's shut off in inactive, unmethylated active. If it's unmethylated and active, the plants have the characteristic floral structure. But if it's methylated and shut off, then it's a developmental gene. They have a different floral structure. And that difference is due to methylation or non-methylation. Epigenetic, and it's heritable. I've worked on a methylation, hypomethylated versus hypermethylated allele of a gene in corn that we first studied in 1985, and I've grown it out and studied it for 20 generations, and that methylation pattern can be retained. It's heritable. So, and I showed you all this stuff. We've gone through all this. Now we're going to fast forward to the last couple of slides that I showed to these kids. And I skipped a couple, rushing through this, but I have I don't care how long it takes with my babble, okay? We won't get to the good science. So the human genetic code, I'm telling 9 to 12-year-olds this, the human genetic code is pretty much the same size as a set of 5 Harry Potter books. I counted this. I determined this mathematically. The information that's in 5 Harry Potter books is the same size of information that's the human genome. So naturally somebody's going to say, how is that possible? How is it possible that it's not far more complicated than that? It's not. So, and you guys probably won't believe me, but it's true. And we go through all this, and I was showing you all these guys. Whoops, I'm going to fast forward. But the last part of this I skipped at the end. Forgot it was there. And that's this. You see this recording studio here? This is what I'm using to explain things to kids. The genomic programming recording studio with control console. With each knob controls the level of a single gene's expression, or say of a transcription factor, that in turn controls networks of genes. And so the structure of a typical gene, you have a protein coding sequence. I don't know if it's too small for you guys to see, about 5 kb on average over, of course, many genes. And then you have 5' of that, a promoter region, which is really the volume control for that gene. And this is how it works. All the genes have volume controls, and it's how you control the volume control for tens, hundreds, or thousands of genes that produce phenotypes, the traits that we're interested in, height, etc. Now, of course, there are traits that are single gene traits, eye color, hair color, many disease mutations. There are important single gene traits. But most of the traits that are really, most of the way we are is determined by the interaction of many genes, or multigenic traits. So you control the volume control on these genes. That's how you end up with the way something looks. So here I am talking to 9 to 12-year-olds. And by the way, I also want to give credit to my wife, who helped me with a lot of this. She's great at doing this stuff, this graphic stuff. So Mother Nature does genomic programming. Here she is. My wife's so good at this stuff. I don't know how she figures all this out. Now watch. Oops, this volume is down again. She changes, she alters the volume control, and the kid's hair goes from blue, from turquoise to brown. That's what's going on. But wait a second. Now this is for kids, remember. Now we humans can do genomic programming as well. Now, that's probably not correct to have science geek. That's like, improper, not politically correct. But and there's a plant. Oh yeah, change the volume control. Oops, goes from green to purple. We can do it too. So anyway, that's a quick review of what I did on Monday. Now let's move on to some actual hardcore and very current science about this phenomenon of epigenetic inheritance and how it's important to what we're all doing, to what we're doing. We're going to talk about non-random transgenerational epigenetic inheritance. I'm working on this myself, and I'll show you examples in the literature, but also examples from my own research. Then we're going to talk about a very important phenomenon down here. See, here's one of those detours. I was going to mention some human inheritance studies, human health studies. Now, I should skip that whole detour. That's what I was thinking about this morning. Skip it. Don't show them that stuff, because it'll use up time. But I'm not going to skip it, because it's too interesting. This is the most important thing, I guess, that we'll talk about today, is paramutation and what I call the Borg process. And then we'll talk about intra versus trans. This is very important to discuss. Intra versus transgenerational stress response. Intra-generationals, you grow in a plant, and there's a huge literature. I don't know how many of you guys are doing plant biology as students today, or hope to be doing plant biology in the future. Research, or really research in any field. Whole bunch of a huge field of literature having to do with how plants respond to stress. Abiotic stress, drought, nutrient stress, heat stress, biotic stress, like what Dr. Balhorn works on. That would be insect predation or diseases, fungal infections of plants. Biotic and abiotic stress. Huge literature, huge field. But almost like 99.9% of all those studies look at how plants respond intragenerationally, within their lifetime. But what about, is there a transgenerational response? We're going to talk about that. That is, does the parent's exposure to some stress alter the programming that's transmitted to their offspring? And if it does, that could be important. We're going to talk about that. And it does, by the way. So, epigenetics of multigenic traits, random versus non-random variation. Key thing here. Traits conferred by non-random processes. Non-random, there's some function, there's been the evolution of a way for plants to sense their environment, respond to it, and transmit new programming for adaptation to that environment to their offspring. There's been evolution of this mechanism. It's non-random, it's a mechanism. There's biology behind it. So we're going to talk about that. Two cases, heritable enhanced tolerance to a drought or nutrient supply, and then this phenomenon of genome-wide paramutation, which I call, trying to be humorous, the Borg process. Does anybody know what I'm referring to? Does anybody know who the Borg are? We will assimilate you. Star Trek, okay. Some people don't like Star Trek. I was a 15-year-old when it came out, and I loved it. But the Borg, of course, was in Star Trek: The Next Generation. We'll go all that stuff. But the fact of the matter is that the Borg process, it's happening. We're going to talk about it. At least I think it is. Okay. Anyway, the Borg process, and then traits, and then we're going to go through this stuff, and then we'll talk about traits conferred by selection for random epigenetic variation. So there's two things going on. There's non-random processes, and then there's random epigenetic variation. I'll give you, since I'm going to babble on and talk about extraneous and irrelevant stuff a lot. So I'll give you the conclusion right now. The rule of thumb for genetic variation, for random genetic, the basis of evolutionary theory up to the present, is selection for allelic variants, sequence differences that impart some favorable trait to an organism, to an individual, and that individual is therefore more reproductively successful. And its progeny represent a larger part of the population in subsequent generations. That's the classic idea of evolution. But it's all based on the rate of random genetic variation, which is a thought of a rule of thumb, one mutation per gene per million individuals. So if you look at a big field of corn, if there's 1 million plants out there, there's a mutation in each and every gene somewhere out in that field. One hit per gene per million individuals. We use these numbers, I use it practically in my own work. By the way, I mostly do genetic research, not epigenetic research. So my whole career, I wanted to say, is based on Mendel, really. We isolate interesting mutations, is what I do for the USDA, and possibly valuable mutations in genes that confer traits of possible value, genetics, classical genetics. And to do this, we increase the background rate of mutation from 1 hit per gene per million individuals, roughly speaking, to 1 hit per 1,000. We increase that rate 1,000-fold. Now we could screen, we could test. Think about it. You're not going to be able to test for some trait, some characteristic, the chemistry of seeds or whatever. You're not going to be able to test 1 million individuals in the lab. That'd take forever. But you can screen 1,000 individuals. This is the basis of mutation genetics. This is what Morgan did with flies, what we've done with plants and others. 1 hit per million. The rate of epigenetic change, that is the rate of change, random change, of a cytosine to a 5-methylcytosine, that's the 5th base, the methylated cytosine, that rate is 4 to 5 orders of magnitude faster than genetic mutation rate. So you're getting a change in any given cytosine in the genome in 1 in every 10 to 100 individuals. And I think that's a much, much higher rate of change. It's 4 to 5 orders of magnitude. It's 1,000 to 10,000 times higher rate of change to go from a cytosine to a 5-methylcytosine than to go to, say, from a cytosine to a thymine or a cytosine to a guanosine, you see. Much, much higher rate of change. Now, that's random epigenetic variation, and those changes can alter changes in gene expression and ultimately in phenotypes. And it's a very high rate of change. You know, I'll tell you quite honestly, after thinking about this field of research over the course of many years, you know, when I look at, I work on corn and barley and stuff like soybeans. When I look at a field of corn, thinking about this epigenetic stuff, I'm thinking, how could it all look so consistent? How could a field of corn look so consistent in growth and phenotype? Because there's so much change going on. We know there is so much change going on. So it's a lot to understand here. So first case we're going to talk about, epigenetics of multigenic traits. This study, heritable alteration, well, here again, transgenerational inheritance of adaptation induced in response to biotic, in this case, abiotic stress. Heritable enhanced tolerance to drought or nutrient supply. What these guys did is they grew rice. Well, two of the cases I'm going to talk about is with rice. That's important because we're not talking about some academic subject that's of great importance intellectually. We're talking about rice, which is one of the 2 or 3, 4 most important crops in the world. Billions of people subsist on rice. Rice is important. What else can I say about it? Anyway, heritable alteration in DNA methylation induced by nitrogen deficiency stress accompanies enhanced tolerance by progenies to the stress in rice. So I'll summarize what these guys did. We're not going to go into all the gruesome details because we don't have the time. I mean, if we just had one paper and spent 3 hours on it, fine, but we have like 57 million papers and you can spend 1 minute on each. But here is a healthy looking rice plant. If you grow this plant with optimal nitrogen, for those not completely familiar with horticulture and plant biology, the 3 big nutrients are nitrogen, phosphorus, and potassium. Nitrogen is a hugely important part of fertilizer and plant growth, as is phosphorus. I work on phosphorus. We'll talk about that later a little bit, maybe. People are very interested in something called nitrogen use efficiency. It's a huge subject in agriculture, and there's been tons of research on it. The number of scientists and agronomists, horticulturalists, that have grown plants and looked at their response to low, medium, and high nitrogen, it's the thousands of studies that have looked at how does a rice plant or a corn plant or any plant, a Arabidopsis plant, respond to limiting levels of nitrogen versus optimal levels of nitrogen. And they've identified many, many genes, we'll talk about this, that are part of that response. So if you grow a rice plant on normal, optimal nitrogen, harvest the seed, and then grow the next generation under reduced nitrogen, the plants grow smaller. They have less nitrogen. You know, their parents were not stressed in any way. You collect, harvest the seed, grow it under reduced nitrogen, plants grow smaller and have lower yield because they have less food. But if you grow the same line of rice under reduced nitrogen, harvest the seed, then grow it in the next generation, the plants grow better with reduced nitrogen. The parent is exposed to stress. That alters the expression of genes that are related to that stress, like, for example, transporter genes, nitrate transporter genes. In my study, I don't have the time to show you all this stuff. In the studies that I've been doing, one of the genes that popped out as showing this transgenerational effect strongly was a classic nitrate transporter gene. Now, how was that nitrate transporter gene first discovered or defined? Scientists were growing plants on low nitrogen, asking which genes are turned on or off, which genes are responding to the stress. Aha, here's a gene, a nitrate transporter gene. And okay, so we've stressed the plants and found out genes which intragenerationally are important to the response to nitrogen. And but it turns out that same gene, if you then look in the next generation, shows that the plant has inherited that change in that gene's expression. So in the next generation, it's expressing the gene at a higher level. Now the plant's growing under reduced nitrogen, it's already prepared for it, you see. I know it sounds weird, but that's really at the heart of what we're talking about. That organisms, I claim, and of course there's many who will say this is nonsense, that, I'm not important. Scientists have shown, that's a better way to say it, scientists have shown that organisms are capable of sensing their environment, responding to it, and transmitting that response to their offspring, which is clearly of benefit, right? It would be, if that phenomenon could happen, if that biology could happen, you'd think there'd be Darwinian in the classic sense of Darwinian evolution, Darwinian selection for that very process. You see, Darwin and Lamarck. If that sort of process can occur, there'd be selection for it. We'd evolve it, because clearly it's going to be helpful for the offspring to be able to live in the world experienced by their parents. It's so simple. But anyway, no, that was meant as a joke. I think it's pretty straightforward. I don't know. What do I know? Anyway, our results thus demonstrate heritability of an acquired adaptive trait in rice, which was accompanied, I give these quotes because they state, and this is in PNAS, Proceedings of the National Academy of Science. This is not a second-rate paper in some obscure journal that wasn't reviewed. This is hardcore science. Our results demonstrate heritability of an acquired adaptive trait in rice, which was accompanied by epigenetic inheritance of modified cytosine methylation patterns, implicating an epigenetic basis underlying the inheritance of an acquired trait in plants. Why are they using these words acquired? Why do they say acquired trait? It's not by accident that they're saying, we're showing inheritance of an acquired trait. It's because of Lamarck. I wanted to show you these guys. This is the Journal of Plant Physiology. This is not PNAS, I'm sorry. Von Wettstein right here, prominent guy at Washington State University. I'm not sure if he's an active faculty member anymore, or I'm sure he's an emeritus or possibly even dead. Okay, so these studies suggest a mechanism for epigenetic adaptation to abiotic stress. So you have abiotic stress or environmental change, induces epigenomic change in generation 0, in the parent plant, that is transmitted to the first generation progeny. A subset of these epigenetic changes that confer adaptation or enhanced fitness are inherited in the second generation progeny, and therefore there is adaptation via induction and inheritance of adaptive epigenomic states, and this continues on in subsequent generations. It's a simple kind of process. And we'll come back to modifications of the simple idea. So, can I take a detour for 4 minutes? I'll only devote 3 minutes to this detour. And I promise you we'll get to the hard stuff, more hard stuff. This is so interesting. Humans and plants both exhibit transgenerational inheritance in response to changing availability of their respective foods. I originally included this example because the previous example with plants was with response to limiting food. In the case of plants, nitrogen. But what these folks have shown is that cardiovascular and diabetes mortality determined by nutrition during parents' and grandparents' slow growth period. And what you don't have to read all these details here. I'll just tell you what they found. That you can read it some other time. It's in the PDF file, etc. What they found was that if they looked at historical records, and they found that if individuals were in the so-called slow growth, that's the 6 years old to 9 years old, 5 to 10 years old, the slow growth period before puberty hits, whenever puberty hits, 11, 12, that period of years. If individuals lived during a time of plenty of food, then their grandchildren had an increased incidence of cardiovascular disease and diabetes as compared with children whose parents were in that slow growth period during a time of famine. So if your grandparent was living in a time of famine, you will have, on average, a reduced incidence of cardiovascular disease and diabetes. Now, I vote that that's an important observation. And again, who am I? Okay. I predict they're going to get the Nobel Prize. Once the scientific community gets over, well, if they're still alive, because it takes so long for the scientific community to get over, paradigms, get to the next level of understanding. But that's a super important finding. Yes?
Just a question. Is it that the grandparents reproduce in that time of famine, or is that...
No, they were children. And if the famine was occurring when they were children, then they grew up and had children. So it's if they were exposed to the stress during their slow growth, the 5 to 10 years old, 5 to 9 years old.
So it's not necessarily if you experience famine after that?
That's not in this study. No. Now, the biology of this is not well understood. You know, how this phenomenon of transgenerational inheritance actually is occurring, that's what people are working on today. But what they're observing, what they're doing is documenting that it occurs. And it gets worse if you guys haven't seen these things. Now, I'm showing you stuff with humans. So I'm claiming that it's the same phenomenon as stuff going on with plants. And that's actually relevant to my basic worldview. Remember that slide where I showed that the little boy and the fish and the fly are really all more similar than they are different? If you've taken statistics, how to use statistics in science, I took a year and a half of statistics. If you take statistics, you learn about something called the null hypothesis. Who here has heard of the null hypothesis? Most everybody. Fantastic. The null hypothesis, to me, is that there's no difference between humans and plants. And people need to prove, if there is a difference, they have to reject the null hypothesis. And now, if you read this literature, if you're interested in this stuff, as I am, people are talking about how plants are uniquely need epigenetic inheritance. Plants uniquely need it because they're sessile organisms. They're stuck in the ground in one spot. They need to be able to adapt because they can't move from Berkeley, California to Portland, Oregon. They're stuck. So they have to be able to adapt because they're sessile organisms. And I'm saying, hup, sounds nice, but that's not good science. Prove to me that there's a difference between mammalian phenomena and plant phenomena. And then I'll believe that there is such a difference. But I just want to show you these guys just to drive home the importance of this. Here's this study, the American Journal of Clinical Nutrition. If your father chewed betel nut, that's a nut that gives you a buzz. If your father chewed betel nut for the buzz, you have a higher risk of type 2 diabetes depending on how much he chewed. It's actually a linear relationship. Amazing. This one's even more shocking. The anthropometry, and that is measurements of things. The height, weight, bone mass. The anthropometry of children and adolescents may be influenced by the prenatal smoking habits of their grandmothers. So here we are arguing in society about how behaviors during our lives can impact our health. Hey, get over it. Let's understand that and recognize, if this is all true, that we had a much bigger problem than that. Because the behavior of your grandparents can determine your own health and well-being. And think how that should affect, I always give the example of air pollution and the example of school lunch programs. Will our decisions in local, state government, and federal government that impact things like school programs and air pollution impact not just the quality of lives of kids today who are experiencing that air pollution and nutritional deficiencies, et cetera. This is actually what I work on. It's what I get paid for, is to work on breeding crops, the genetics of crops for nutritional quality. So I've worked on international nutrition issues like zinc nutrition, malnutrition worldwide, that kind of thing. And think how our decisions today, we need to take into account they can impact not just us, but our children and grandchildren. I think that's a different perspective from my point of view. So why do I go on about these human inheritance studies? Because for me, the null hypothesis is that basic mechanism of inheritance in humans and plants, both Mendelian and epigenetic, are similar. Prove to me they're different. I'm going to assume they're similar until somebody shows me otherwise. So when Mendel was studying peas, didn't the laws that he discovered apply to humans? They did. So I think the laws of epigenetics also probably apply both ways. So we'll get on to this next important subject. And I am on schedule. Sort of. I hope I'm not overwhelming you guys with too much stuff. If it's just too much stuff, just raise your hand and say, this is too much stuff. Is this too much stuff? It's not too much stuff, right? So anyway, I came across, I wanted to mention this. This is so interesting, but now I'm taking too much time. This person here, Marcus Pembry, is a physician. And he makes an interesting point. He's writing a review of a book and a paper by two scientists, Jablonka and Lamb, about these questions that I'm talking about today, about epigenetic inheritance. And And that they wrote a book, these persons wrote several papers in a book back in like the 90s that were very influential and very forward-looking. And he says, Jablonka and Lamb went beyond their title, Inheritance of Acquired Epigenetic Variations, to argue for a role of epigenetic inheritance in evolution. In other words, not just look at how this might play a role within a couple of generations, but the big picture story, evolution, the big story. But I guess it's because nothing in biology makes sense to many biologists, except in the light of evolution, right? That's what biologists are thinking about. It is what biologists, particularly theoretical biologists, do. Think about how does this biology, evolution, that's the big topic. But for medicine and epidemiology, and I want to say he's a doctor. When he's talking about medicine and epidemiology, he's talking about applied biology. That is actually exactly the same as what a plant biologist does. He's talking about the health of children, of offspring. I'm talking about the performance of plants. It's the same thing. He says, we don't have to work on such a distant evolutionary horizon, i.e., we don't have to think about the long-term big picture above evolution. One has only to make sense in terms of family, public health, and well-being. Indeed, when in the 1990s, a colleague said to him, inheritance of acquired epigenetic mark could only have a trivial influence on evolution, he replied, I'm a physician. I'm not worrying about, evolution and big picture stories. All I'm concerned about is there's a person in my office, in our clinic, who is pre-diabetic. And part of the explanation for that could be his parents' and grandparents' habits. That's important to me as a doctor. I'll let you guys worry about the deep philosophical or long-term issues. So let's go on to an aspect of how this actually works. Get into the nitty-gritty here, the actual mechanism. On the left here is your standard Mendelian segregation, Mendel's first law of segregation. If you cross a big B by a little b, you get a heterozygote, big B, little b. God, I can only tell you how many times I've done exactly this in my work. And then self-pollinate it and see segregation, Mendel's first law, of these alleles in the progeny. Meiosis, segregation, and independent assortment. Paramutation breaks Mendel's law. It's the first case of an observed inheritance which broke Mendel's law, this first law. This is the big B, just like this guy. But over here, if you see it's B prime, it's a different kind of allele. And we're going to talk about this allele. Now you cross these two, you get a heterozygote, big B / B prime, but the B prime allele, big B, big prime, and we're going to talk how this works, the B prime allele converts the big B allele to a B prime allele. Now in the F1, you've got, instead of a heterozygote, you have B prime, B prime. The B prime allele, it's an epiallele, I'll tell you how it works in a moment, converts the normal allele to an epiallele. And now all the progeny of B prime, the Borg process. The B prime, I will assimilate you. There is no resistance. Do not try to resist. All resistance is futile, that's the line. All resistance is futile. So, this is how it works. I don't know if you guys could see it, but this is a stretch of a chromosome in corn, about 100 kilobases. Here's that B gene. Over here, 100 kilobases away, is a very short, like 20 base pair sequence. It's not next to the gene, it's 100 kilobases away. And this is the normal allele. And you see it's green. What this author, this is non-methylated active enhancer element. It's not methylated, and it's making little short RNAs which go and turn on the gene, and the gene is expressed normally. Over here is the B prime. In this case, it's the same B gene, but that enhancer is methylated and therefore shut off. It is not making its little RNAs, and the gene is not transcriptionally active. Now, if you cross these two to make this heterozygote, the methylated allele over here, through a complex process that we can't talk about today, ends up methylating the enhancer that is unmethylated. It gets methylated. It converts the unmethylated active enhancer to a methylated silenced enhancer in a cell, in the nucleus. And now both alleles are B prime, are methylated, silenced alleles. And therefore all the progeny are silenced, have that allele silenced. This is called paramutation. I'll spend the rest of the time just talking about this one phenomenon. It's so relevant. And we'll quit with plenty of time to spare, I promise you. I'm not going to make any promises. But anyway, this is so relevant because this was discovered, R.A. Brink, the person who actually discovered this phenomenon first, is a guy by the name of R.A. Brink at the University of Wisconsin. I actually had a desk, his office was in the basement where as a postdoc I was, and he was a great, great scientist. He discovered this phenomenon in corn, and from 1950 to 1990, really from 1950 to 2000, for like 50 years, there was only 4 examples of this phenomenon, all in corn, all with anthocyanin pigmentation. Because the reason why they were observed is because it's easy to see patterns of inheritance if it's pigmentation of a plant. You could just grow out plants and you could just see it, or in the kernels. Very easy to discover. Good model system, in other words. And for 50 years, there was just these few examples. The 5th example turned out to be a gene that I had been working on. It wasn't in the anthocyanin pathway. It's in an inositol phosphate, a basic housekeeping carbon metabolism pathway, not a pigment pathway. But we observed it, colleagues of mine. And I'm thinking, oh, wow, that's great. Now that's the 5th example. And it's the first case outside of this pigment pathway. Hey, this is starting to look like this might be something real and important. That feeling didn't last long because within just a year or two of that, it turned out that Mendelian and this study, this is just one of several that came out around the same time, but this was the first one, I think. Mendelian and non-Mendelian regulation of gene expression in maize, and what they found was, we found 145 genes that show patterns of expression reminiscent of paramutation, such that all the progeny had expression levels similar to one of the 2 parents. That's what we're talking about here. That all the progeny of some cross, the expression of a gene is similar to one or the other parent. There's no segregation. Everybody follow me so far? If this wasn't potentially important, I wouldn't be bugging you about it. So all the progeny show only one parent's phenotype. No segregation. Okay. Several papers came out showing that this phenomenon of paramutation is current throughout the genome. The way you study it is you cross 2 individuals that are very different. You get the F1, then you let them self and get all the progeny. And you could look at now the expression of all the genes, with RNA-seq, with modern methods. And you say, oh my God, what we're seeing here is not segregation of levels of expression, of gene expression. We're seeing often, not just occasionally, but often, the expression is the expression level of one or the other parent. Well, how could that possibly be? Here's a paper that came out, freakish and rare, or the tip of the iceberg. Is this phenomenon freakish or rare? And I'll tell you, I was thinking about this and how I can explain to you guys. Hey, if I'm studying something and I have a choice of studying something that is freakish and rare, or typical and normal, I'm going for the freakish and rare, okay? So what they found is that in tomato, there's tons of paramutation-like phenomena going on. Thousands of genes show this paramutation phenomenon. That is a level of expression, which is what biology is all about, right? Reflects either one or the other parent. There's no Mendelian segregation. So now we get to a paper which I might spend the rest of my time talking about, because I was mentioning it to Daniel, Dr. Bellhorn. This came out not too long ago, 2017. Epigenetic segregation by genome-wide loss of heterozygosity and its association with salt tolerance in rice. So what could be more important than drought or salt tolerance, stress tolerance in something like rice? Billions of people subsist on rice. We here in America, we're lucky, we're fortunate, we're so rich. But out there in the real world, rice, if you ever have an opportunity to visit Asia, you'll find that they eat a lot of rice. You know, it's an important component of their daily diet. It's important. Now, let me explain what these guys did. What I'm talking about. In a breeding effort to develop salt-tolerant rice varieties, progenies were derived from four crosses between salt or drought-tolerant lines. So they're crossing a line that's drought-tolerant and another line that's not tolerant, that has good yields. They're making these crosses. And they're going to study them with traditional methods. They're going to look at, they're going to then get the progeny from these crosses, 422 salt-tolerant plants. They get the progeny and look for inheritance of allelic variation, so-called quantitative trait loci, QTLs. That is genetic variation, sequence variation in alleles that are associated with tolerance to drought or salt. It's really the same thing. And the genetic characterization of these 422 F3 progeny and 318 random F2 plants, just random controls that aren't exposed to stress, just random plants, lead to three interesting discoveries. Salt stress can induce genome-wide epigenetic segregation, they're calling it ES, characterized by complete loss, okay, pay attention, complete loss of heterozygosity, or loss of heterozygosity, LOH. Loss of heterozygosity and nearly complete loss of an allele, LOA, loss of an allele, in the F3. And 25% of the stress-induced loci, ES loci, epigenetic loci, were transgenerational and inherited from their salt and drought-selected parents. What they're finding, by the way, you know, you could find in the literature any old garbage, right? You know, you could find studies that, you know, say anything. So you're always looking for, is this, you know, authentic stuff? Is this good stuff? So this is work from the International Rice Research Institute in Los Baños, Philippines. I've been there. It's a prominent international rice research and breeding organization, part of the international network of agricultural research. Top of the line people. And the other guys in this study are from China's Academy of Agricultural Sciences in Beijing. China's #1 agricultural research center. Happens to be in Beijing. And what they're finding is that it took them several years. I was familiar with this work before it was published. And they had trouble getting it published because they didn't have the proper controls. These 318 random F2 plants, they didn't have the proper controls initially. But then they redid it and looked at these controls. And what they found is that the salt and drought-tolerant progenies that they're breeding were not due to the inheritance of genetic alleles, sequence-differing alleles. They were due to the fact that the parents were exposed to drought and salt stress. These were epigenetic alleles induced by their exposure to stress. So, and this complete loss of heterozygosity, loss of heterozygosity, or loss of an allele. What are they talking about? Paramutation. They actually use the word paramutation in the paper if you read it once. But that's what it is. Whoops. I thought I had inserted. Where'd it go? Huh. I had inserted this. This. Loss of heterozygosity. Loss of an allele. This parental allele, big B, is lost. All the progeny have the B prime allele. Loss of heterozygosity, loss of parental allele. That's what these guys are observing. And these guys are this is a reputable group of scientists. And that's what they're observing. Now, here's one of the ironies of life. I started talking about this in lectures here and elsewhere, like 4 or 5 years ago. And as I mentioned on Monday, at that time it seemed like a stretch. You know, paramutation playing a role, the Borg process. Oh, make it sound humorous, then people kind of think it's interesting, you know. It's the Borg process. But this is a case of it, you know. This is it happening in the real world with rice, having to do with drought stress. This is important, I'd like to think. So, let's revisit how studies are revealing mechanisms of heritable epigenetic adaptation, and let's include genome-wide paramutation. Abiotic stress or environmental change induces large-scale epigenomic change. A subset of epigenetic changes that confer adaptation or enhance fitness are inherited in the next generation. But in addition, this is the model I showed before, in addition, genome-wide paramutation will fix, it fixes adaptive epigenome change quickly. It will use the newly induced adaptive epialleles as substrate to convert original non-adaptive alleles to the adaptive epiallelic version, i.e. the Borg process. That's what I call it. Probably not a perfect example, but that's true. I wanted to mention, and I'll do it briefly because we're going to have to go soon, that if you have stress, this is what this paper talks about, if stress induces changes in gene expression, they're not all whether they're adaptive or not, that word adaptive is tricky. Certain changes in gene expression may be adaptive in a given environment. That same change may be maladaptive in a given environment. The same stress-induced change. What they're talking about in this paper is that drought response studies. If you expose plants to severe drought, there are changes in gene expression which are transmitted to offspring, but those changes provide adaptation to severe drought. But if you grow those plants under moderate drought, those same changes in gene expression are maladaptive. So a way to think about it, if you know your plant biology, if you have severe drought, the stomata close, and that closure of stomata is adaptive under severe drought. But now if you're growing those plants in the moderate drought, severely closed stomata are not adaptive. They have some water. They need to have their stomata open a little bit to do transpiration, etc. Not all changes in gene expression are adaptive. It's context, environment dependent. And that's actually, I mention it because it's important, but also it impacts how we do this research. So now, I'm going to skip over because we are going to run out of time. But I'll summarize this quickly by saying that so many studies have looked at stress response, abiotic stress response, particularly in intragenerationally, within the growth of a plant. And what this review article is talking about is that it is striking that wait a second, up here. A survey of PubMed database reveals that the great majority of epigenetic papers have intra rather than inter or trans generational focus. Give you an example out of my own work to bring this home, the whole field of study home. We have a few minutes. I've been working on phosphorus biology in plants for many years. And there's a huge field of research having to do with how plants sense the amount of phosphate in their environment. It's another one of the big three, N, P, and K. Phosphorus is critically important to organisms. They have to have a certain level of phosphorus to grow, as do plants. And there's a huge field of literature that looks at if you stress a plant by giving it too little phosphate, how does it respond to that? It's a huge field of research. Not just with plants, with yeast, microorganisms, it's a huge field of research. Now, I say, this is actually from a grant I wrote back in 2014. It is striking that despite the very large number of studies that comprise this field of phosphate sensing and phosphate stress response, not one of the studies, not one, when I wrote this, has taken the next step to describe the transgenerational transmission of this response. So you grow plants under phosphate stress, you look at how it behaves. Not one of all those studies, including studies that I've done, harvested the seed from those plants, grew them in the next generation, and asked, do any of those responses, intra-generational responses, do we see that in the next generation? The answer is, yes, we do. And, it's going to get late. Here's an example of one of those studies. A genome-wide transcriptional analysis using Arabidopsis thaliana, which is a model organism in plant biology, chips determine plant responses to phosphate depth. This is what these guys did. They grow, and this is excellent work, Misson et al. Grew these Arabidopsis plants, stressed them for phosphate, and looked for the genes that were expressed differently in response to that phosphate stress. And so we did an experiment, which we haven't published yet, which we really need to publish. Have a simple hypothesis. Let's do the same thing. In this case with barley. which is very easy, a great model organism. Let's do the same thing. We'll grow the barley plants on the phosphate stress. Let's harvest the seed and take seed from stressed and non-stressed plants, plants whose only difference, they're same cultivar, same line of barley, they only differ by whether their parent was exposed to stress or not. Let's then grow those two progeny, which are identical in every way except the parent was grown under full phosphorus or reduced phosphorus, grow them in the next generation under reduced phosphorus, and ask how many of the genes in this paper, Bisson et al., that have been identified via an intragenerational study of stress response, how many of those genes show transgenerational response? Or that the progeny of the parent that was stressed has a higher level of expression in the same environment as the progeny whose parent wasn't stressed. Okay, everybody understand the idea? The test? Simple test. We're not saying let's discover genes that have stress response roles. We're taking the genes from their paper, the barley orthologs of these genes. Saying, okay, how many of those genes show either intra or transgenerational expression? Now, what do you guys think the result is? Do you think 1% of the genes that show intra-generational response also show transgenerational response? I should say, do you think 0, none of the genes show this? 1%, 5%, 10%, 50%? What do you guys think? Let's say 0%. Obviously, that's not true. 5%? Anybody? 5% of all the genes that they just looked at, intra, showed trans. 10%? Okay, I'll give you the answer. It's somewhere between 10 and 50% of all the genes. Now, and I'll show you the results. This is a huge field of work, this type of work. It's not just phosphate response, drought response, salt stress, drought stress, heat stress. This is a huge field of research. And yet, so few have thought about intra versus transgenerational adaptation. So, another example of what his guy's talking about. Multiple exposures to drought train transcriptional responses in Arabidopsis. This is during, what they're doing is they're taking Arabidopsis plants, exposing them to drought, and then removing them from drought, giving them full water, and then later on in their life, reintroducing drought in the lifetime of the plant. And they find that if they treat them as young plants, expose them to drought, that they'll be adaptive. Later on in their life, they'll do better, their same life. But now we found that there's transgenerational training going on. That's the point. That there's transgenerational training going on. And here's a barley plant. Yeah, we're doing okay. Here's a barley plant growing full phosphorus and nitrogen. Here's one growing, you see how much smaller it is? And we harvest the seed from these two plants and grow them in the next generation side by side under moderate stress. The only difference in these plants in the next generation were whether the parent was not stressed or stressed. And when we do RNA-seq, and I need to publish these results, if the parental generation for this particular gene happens to be a gene in the pathways that I've worked on for many years, the inositol phosphate pathways, we see 141 counts in the plants that are grown under full nutrition versus 240 in the parents. So in the plants grown under low phosphate and nitrogen and potassium. So a doubling of expression of this gene in this plant versus this plant. Then we harvest the seed and grow them in the next generation under the similar level of nutrition, side by side, same level of moderate reduction of nutrition. And we see a big increase in the progeny of this guy versus the progeny of that guy when grown in the same environment. And we see this difference, this transgenerational adaptation, in somewhere between 10 and 50% of all genes that are previously shown to show stress response. So, to conclude from all that, with 5 minutes to spare, the fact is that that you can see in plants this phenomenon of transgenerational adaptation. And the question, if you recall my very first slide, there's this question of not just simply observing this phenomenon, but quantitating it. Quantitative biology. How many genes show this effect? That's what's important. To get, well, it's not the only thing that's important, but that's what I'm getting at. They're getting a quantitative handle. So if you work in this field, if you're one of these prominent scientists that work on stress response in plants, you know, you're studying a phenomenon. If you could show that there's a transgenerational adaptation effect, isn't that important in how plants adapt to their environment? That's the bottom line point. That's the conclusion. And this shows that result, and I'll spare you all the hard details. So that's, actually, look at that. Holy mackerel. I still have 5 minutes. And it's lecture 3. Oh my God. Sorry guys, you can't go anywhere yet. I'm going to show you the hardcore details then. So this is the hypothesis test. It's a kind of so-called candidate gene approach, which is how many of these genes do we observe this phenomenon? And here's our answer. If you look down here, hypothesis test result. Whoops, I cut this off. Look at that. Hypothesis test result. Sorry about that. Negative, positive, positive, negative, negative, positive. These are individual genes that where you look at the differential expression in the parents versus the differential expression in the offspring. And like I said, we see transmitted, don't worry about it, you'll get confused if you start looking at all the numbers. What we're looking at when we talk about differential expression, log base 2, is the level of expression in a stress plant divided by the level of expression in the non-stress plant, in the parental generation versus the offspring. And what we find is that we see differential expression based on parental exposure. Now, I'd love to continue studying this. I'd like to take this out to 3, 4, 5 generations. I'd like to compare this to see whether this expression, whether there are remnants of this expression even in the absence of stress. Presence versus absence. There's a lot of stuff to look at to continue this. But you know what? It's happening. And it's happening in plants. I'll leave you with one last thought. Let's say you're studying, you're breeding, you're a scientist, a breeder, working on disease response in plants. I go on about this one. Disease response. There's important diseases, things like stem rusts. There's important diseases of cereals and legumes. And you're studying, say, disease response. And my colleagues are studying disease response, are trying to breed rust-resistant wheat lines. There's a lot of money for this, to breed wheats that are resistant to rust. How do they do this? They send all their lines, their rust-tolerant lines, disease-tolerant lines, and disease-resistant lines to a nursery in Uganda, because at that site, there's a lot of this disease. So, okay, we could see how this line is doing relative to that line, right? They're all exposed to this disease. So I'm saying, guys, if you grow them under that, the only way you could do that is by growing them under that disease. But if you do that and you're looking for genetic inheritance of resistance to that disease, but they're all being exposed to that disease. And that's a form of biotic stress, and that can induce response, which can be transmitted to offspring. Well, I got arguments about this. And literally just a couple of weeks ago, a technician working on the project says, Victor, I hate to say this, but we think we see what you're talking about. That all the progeny that come from that nursery, whether that line had been identified as a disease-resistant line versus a disease-susceptible line, but all the progeny show, he says, we think we see that all the progeny coming out of that nursery show a little bit elevated resistance in the next generation when they grow them in the greenhouse in the absence of the, you see? And so, there's a lot of research having to do with plant disease, and breeding for disease tolerance. Anyway, I'm glad I got through my second lecture in time.